Hello, everyone, and welcome to today's webinar. Easily upgrade your flow cytometry data with brightfield images using the Attune CytPix Flow Cytometer. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Cora Chadick, Technical Director, Flow Cytometry at Amsterdam UMC. Cora, you may now begin your presentation.
Hello and welcome. My name is Cora Chadick. I am the Technical Director at the Amsterdam University Medical Center, location UMC, in the Molecular Cell Biology and Immunology Department. We have a nice wide array of instrumentation here in our core facility at the south of Amsterdam. We have mass cytometers, standard fluorescence flow cytometers, spectral cytometers, sorters. But what I've been really excited about these days is our opportunity to demo and do some testing on the CytPix, the new imaging cytometer from Thermo Fisher. So this is built on top of the Attune NXT acoustic flow cytometer. And I was really excited to be able to add this imaging function to an Attune because our Attune is one of the workhorses in the lab. The fluidics are very, very stable. It just keeps running. It's able to run really large volumes. So the sample line is really easy to clean to reduce sample carryover. I just love this instrument. We have a skin lab here that runs all of these very large cells that are adherent cell lines, and we can just clean out the system really nicely. So now I'm going to take you over to the software and show you the real power behind the system. Okay, so here we are at the software. This is how after you've opened the software and run a startup and a performance test, how it would look. You see these modules for creating or doing these QC tests, a new experiment, importing an old experiment, creating a new experiment from template. I want to start a new experiment. I could click on new experiment, but I'm going to use a template that I know I want to use for this demo. I will open that up, and you'll see that I already have some plots that I like to start out with, our standard forward and side scatter, and then a couple of doublet plots. I will be running 3 different colors on the system today. So 2 antibodies and a live dead stain. One will be in the first detector from our blue laser, and I don't like taking the height or the width off of our color parameters. And then also the first detector on our red laser for a Alexa Fluor 647, and then our live dead blue stain. And it's just easy as that to select the parameters that you would actually like to collect. And then once I come under this new tab, you can see we now are able to find the voltages and that only the channels that we've selected are available. When I can also play with the threshold if you want to threshold out debris, and all of your other advanced settings like area scaling and window extension can also be found here. So the next part I like to show is the fact that we also have these set volumes here, or set flow rates here, and we can use any variable volume between 40 microliters to 4 milliliters. So first I'm going to actually grab an unstained here. And maybe also add some dot plots for each of our color parameters in our nice workspace. Could also choose density, precedence density, and I will stay away from histograms for now. And I am in sample 1, as I would like to be. These windows are all modular too. I can kind of place them where I like. So right now I'm going to bring my experiment browser over here so I can have a better look at my sample and my color channels. I will go ahead and click run so I can start previewing what the sample looks like. And there's our forward and side scatter. I would say our forward scatter is a bit too low for these cells. And the really nice thing about the software too is every time you change a voltage, you automatically get this refresh in your data view. And side scatter looks good enough. We're looking at PBMCs today. So you can see that these are nicely on scale for forward and side scatter. I think I would like this plot to actually be a density plot, and I'm going to change the resolution. It really is just as easy as that in this customization panel, or in this customization window. Now I need to look at my color parameters. So here's my live dead channel, and then my CD3 and a CD14. So now that I have my forward and side scatter set, I don't think I need to change the area scaling at all. So I will go ahead and stop. This is a syringe-driven system, so it has taken up 25 microliters, and I am able to recover a portion of that if I like, either into the same tube or into a different tube. I'm going to recover into the same tube. Then I get my sample back. And I am able to retain that remaining 71 microliters for the most part. There's a bit of depth volume there too. So now I want to check my full stain control and make sure our positive signal is also on scale. Since I'm drawing 100 microliters and running at 25 microliters per minute, I have about 4 minutes to do all of this setup, which should be plenty of time for 3 parameters. So here we can see that we have really nice CD14 staining, but our CD3 doesn't seem to be super strong, but we also have nice clear live dead staining here. I think I want to get a little bit better separation on the live dead, and maybe I can do the same with the CD14, which is on our red channel. I'll try 450, 450 and 450. That sounds nice. Oh yeah, that looks a little bit better. Okay, I have my setup here. I also know that I've looked at these samples earlier and there's not great separation with the CD3. I could zoom in here, maybe change the resolution and get a little bit better view of that, but I'm not really sure about that shoulder. So I'm going to go ahead and stop this again and recover my sample again. And lower the tube. And let the sample line rinse. So now this is everything that I think most people are used to seeing in a standard fluorescence flow cytometer. And so, yeah, now we get on to the really cool part. I've already done a bit of calibration beforehand. You can see now I can come down to these image capture settings. And so this is something new that I think most of us at a flow cytometer are not used to seeing. Now we have some image capture settings that we'll need to play with. So I know earlier I found our focal plane pretty well. This is our CD14 only stain. But I want to find the correct focus for our flow rate and for the sample. And here you can already see that we have some cells. I know some of you are probably already saying ooh and ah and wow at this point, but of course it'll get even better once I find the correct focus for these cells. So I'm going to run some more sample again, imaging every event on all events. Not set any image gates here so far. So as you saw with the last images, we are quite out of focus here. So I'm going to play in increments of 10 with our focus to see if it gets better or worse. I think we can all agree that that is worse. Go go for 57. Okay, much closer. And if your sample rate is fast enough, you can usually see some preview, so you can see those are actually getting a little more out of focus. So I'm going to continue further into this focal plane. And if you weren't saying wow before, I know you're saying wow now. This was just during the very first training session, my jaw dropped. This region of interest on the camera. So this is our entire camera chip represented over here. We can select different areas on our camera chip because it's a flow cytometer. The cells might not always be in the exact same spot every single time. And so we just need to have a little bit of flexibility in that positioning, especially for our illumination setting. So if I want to maybe change the contrast of these images a little bit more, I can also turn up or down the illumination and also find different points on the camera chip that will maybe give me a little bit more or less contrast in our imaging here. If I try 80, that's going to be much darker. Let's see, much heavier shadows on these cells. So these are illuminated with 405 nanometer laser. Let's go back up. Oh, way too bright. Yeah, completely saturated. So I think we were already in maybe our sweet spot there. Whoop, nope. 45. There we go. 48. Okay, there we go again. I forgot momentarily where our range was. I inverted our numbers here. Went from 84 to 48 instead. But we are back in action. It goes that quickly. It is really that fast after you use the system a few times to find a nice focal plane that you like and a nice imaging area that you like as well. Playing with the focus just a little bit more, playing around a little bit to see if I might like one spot a little bit better than another. So I think on average, most of our images here that you can see in the image gallery are maybe skewing up and to the right. So I will simply move our region of interest for image capture up and to the right. So now we're a lot more centered with these cells. Okay, so I think I'm pretty happy with these settings. Maybe I could play around a little bit more, but for demo purposes, I also want to share with you a few other experiments that we've done, especially with some larger cell types where this becomes really exciting. But just to show a little bit more about the capabilities of the system, you have all of your normal stop options that you would have on a flow cytometer. So let's say I want to record 50,000 on our P1 gate, on our R1 gate, I should say. There we go. And I only want to record every 10 events out of that 50,000. And I only want to record events that actually fall in that R1 gate because I'm not particularly interested in imaging debris or what we assume is debris. Okay, so if I clear this, I will go ahead and record. I will likely stop this early because we're running at 213 events per second, and I don't think I actually want to wait for that 50,000 events. So now you'll see these images are coming up a bit less frequently, and that's because I have our image capture settings to that frequency of every 10 events. If you are collecting hundreds of thousands or millions of cells, you can also have your total images be limited as well. And it also gives you a nice estimate of how much of your hard disk you'll actually be taking up with those images. All right, I will stop early again. I like having 10,000. That's enough. Okay, so we're at 10,000. We imaged almost 1,000 cells, which makes sense if we're taking one image for every 10 cells that actually fall in that R1 gate. So that's how every day when I'm setting up this the experiments on the system, it is about 15 minutes from initially powering everything on to the moment that I am able to capture images. I think everyone in flow cytometry always enjoys the instant gratification in results that a flow cytometer can give. This just takes it to the next level, really. Another nice thing about this system, since it is syringe-driven sample acquisition, we can also, just to show a little bit more of a capability of the instrument, we can also see your concentration in microliters. So this particular sample in our R1 gate, it was almost 500 cells per microliter. So it's nice to, you don't necessarily need to use counting beads to find your cell concentration. So that's how quick it is to actually set up an experiment. And so this is kind of our, if you were on a cooking show, this is the point where we've assembled everything and put it in the oven. And then, of course, this would be our dish that we have pulled out of the oven, since I called it demo pre-baked. So this will look very familiar, but in this system or in this layout, I can run our full stain sample, even though I have switched experiments. Since the focus of the camera changes from day to day, our image capture settings have still retained, even though we've fully changed experiments. So I can, let's slow down our flow rate. Let's do 100 microliters per minute this time, and have a little bit more of a reasonable amount of events that we want to capture in this case. And we can create our R1 gate again. The settings in this experiment are the same as the ones that you saw in the previous experiment, but I've already run all of our single stain controls, and we have compensation applied to this experiment. I can easily rename my sample, just call this full stain. And what do we want to do this time? Let's stick with that R1 gate, and let's image every 5 events. And this time I'm going to go with this 5,000 for our target. Actually, I'm going to image every event and have our total images target be 5,000. Come back to my collection panel and hit record. There's our cells again. See, we're back to the same settings. And I think we can all start recognizing some different cell types in our PBMC mix here. So we met our stop options, and we've got 5,000 images because we asked to have 5,000 images, and all of them are loaded into this image view gallery over here. And you can see they're mostly singlets, but we can also see our, if I apply R1 here, let's take a look at our CD14 positive cells. And you'll see in a moment why I keep changing these gates from red view to kind of more cool colors rather than warm colors. So now I can select that R2 region. And here you go. These should mostly be macrophages. And if we are curious as to where our macrophages lie on our dot plot, I think we should expect them to have a higher forward and side scatter, as we would all expect. And if I go to a dot plot, change the resolution, we can also come to our image gallery, select a few images over here. If I hold the control button, right click and show selected images on plots. There we go. Right up in that macrophage cloud, exactly where we would expect them to be. So that's how you're able to view images. There's also a measurement tool clear all images here, where you can actually see the size of each cell. And I am able to find the volume of our cell, and we can, or area of the cell, apologies. And we can, of course, back calculate to find the average diameter. And you also know the number of pixels as well. So measuring the cell size is quite easy on the system. And at this point, I wanted to show off some other experiments that we've been able to run here. I will just bring this window over here and tether it to this section of our screen. This one is pretty cool. It's we're looking at immature and mature dendritic cells. So this is another user. I wasn't actively here when these were being acquired, but the images, as you can see, are quite nice. So these are our immature DCs. And then I can come over to select the same sample with the same staining, our mature DCs. Look at that big hairball. So cool. Maybe select a marker here. Let's look at this R6 gate. Yeah. Just wow. Just wow. And kind of one of the very first things that we thought of doing at the system was looking at really, really rare populations, so really infrequent cell populations. This is not the day here. This is the one I wanted to share with you all. So in this sample, we've got PBMCs with a spike in of 10 cells from a tumor cell line. And normally there's quite a few supposed false positives because our healthy donors often have double positive in this population where we would expect essentially nothing to be there. But once we come to our imaging cytometer, we can see that, in fact, most of those positives are, in fact, debris and other cells that or doublets, or who knows what is in there. Also, another nice big reveal is how dirty our sample prep can be sometimes once we are able to look at all these cells in suspension. So there should be about 10 tumor cells in here. I'm just going to come through on this image gallery and select what I suppose to be these tumor cells. So we had 1, 2, 3, 4, 5, 6, 7, 8. So 8 out of 10, pretty good in my opinion. Now I have them selected and show them on the plot, and there are my 8 cells. So once we actually use our standard doublet gating, as you might normally do, or maybe you'll just use this R2 instead, and I actually select the R4 gate, you can see that we actually only have 3 cells there, but when we looked at all of our images, we actually had 8. So we're missing out on 5 possible cells that we would possibly identify as double positive for that population. And if we come to a sample now where we've spiked in 50 of these cells that we expect to be double positive on these 2 channels here, you look at your R4 gate again, we have 14 events here. However, when you come to your R6, I won't belabor the point, but we did go through and count these, and I think we had about 48 cells, including lots of debris. This has also helped us become a little bit more intelligent in our gating. I know to all of you, it's quite obvious that this is our cluster of positive cells. However, once we start going into actual patient samples and we are not just looking at tumor cell lines that are nice and clear that you have a single cell, this will become much more muddy. And so we're in the middle of that project, and I'm really excited to see how that develops. That is all I have to share with you today on the system. I have thoroughly enjoyed using this for the past 3 months, and we'll continue to use it as much as possible. We're training new users on this system, so I think we're going to be getting a lot more interesting data out of here. And I just want to say a special thank you to the team at Thermo Fisher that has let us demo this system and built this wonderful instrument that I've really enjoyed using. And thank you for joining me today.
you, Cora, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, are there any specific steps you have to take for sample preparation?
So this is Cora speaking now. There are no special steps that you need to take for sample preparation. In fact, what I have regularly used this instrument in the lab for lately as we're developing our protocols and how we want to actually train researchers and users on the system is using the system to do a small validation with fixation of samples. So we essentially, when I see perhaps what I suspect to be poor fixation at another analyzer system, I will then take them over to the CytPix just to have a quick view of the cells and see if there is any compromised fixation of the samples. So no, there's nothing special that you have to do with these samples to take these images.
Great, thank you. Next question, what type of cells can be analyzed on CytPix?
Pretty much any type of cell that you would analyze in a conventional flow cytometer. So bacteria, yeast, mammalian cells, anything that you can collect your traditional scatter and fluorescence data from, you can also analyze on the system.
Thank you. It looks like we have time for one more question. How easy is it to transition from a conventional analyzer to CytPix?
Oh, very easy. It's not even really a transition. Just the extra consideration is making sure that maybe you have a little more storage space for the images, because you're not only acquiring those FCS files, but you also have images attached to those FCS files. But other than that, there's no special consideration outside of what you would normally need to do with a conventional flow cytometer. So making sure that you have bandpass filters in front of your PMTs that actually select the light from your target fluorophore. So exactly the way you would design a panel on any other system.
Okay, great. Thank you. It looks like we have time for maybe one more question. I would like to know what next interesting experiments they are about to plan to test new limits of the CytPix. If you have any information on that.
I think that is something that would be best handled by our collaborators at Thermo Fisher.
Okay, great. Well, thank you. Cora, do you have any final comments for our audience?
No, just thank you for having me. And like I said in the video, I'm excited to continue using the system.
Great. Well, thank you again, Cora, for your time today. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.